The previous video showed how the HSQC experiment uses single bond coupling between carbon and protons to produce cross peaks that connect the chemical shifts in both spectra. These cross peaks for single bond correlations indicate that two nuclei share a single bond partnership. The heteronuclear multiple bond correlation or HMBC experiment uses coupling over more than one bond to connect remote proton-carbon pairs. Typically, these couplings are over 2 and 3 bonds, but there are some rare cases when 4 and even 5 bond couplings can be seen. As we will see during the interpretation, these correlations can help construct a complete assignment of both the C13 and proton spectra for known structures, or help build structural fragments for unknown samples. The 2 and 3 bond coupling between proton and carbon vary from about 1 to 20 Hz. They depend on factors that include the hybridization of the carbon nuclei and other elements that are in the structure. In many cases the 3-bond coupling can be larger than the 2-bond coupling in the same structure, and we will see this later. Like HSQC, HMBC is an indirectly detected experiment and offers better sensitivity than the similar directly detected experiment. However, one important factor to understand before running a HMBC experiment is that the intensity of the signals is spread out over several cross peaks compared to the HSQC, which has only one correlation for each proton resonance. Therefore, the HMBC experiment could take significantly longer time to run than the HSQC experiment. A rule of thumb is to run at least 4 times more scans for the HMBC compared to the HSQC experiment. However, this can vary depending on the sample concentration. ICON NMR has 2 gradient-based HMBC experiments for C13 long-range coupling. HMBC GP is the recommended experiment to choose. The HMBC, ET, GP, L3, ND experiment is supposed to suppress the remaining single bond intensity. However, the effectiveness of the filter depends greatly on the magnitude of the single bond coupling constant, and does not work well for all carbons in a structure. There is also an experiment that is set up to observe long-range coupling to nitrogen-15. This experiment is more difficult to run because nitrogen-15 is only about 0.4% naturally abundant, and will require a much longer collection time than the carbon-13 experiments. The HMBC options are composite experiments and will first run a proton spectrum and optimize the window of the 2D run. The sample used for this demo is the same 25 mg ibuprofen in chloroform used for the HSQC experiment. While the defaults for the proton experiment are okay. We will increase the number of scans for the HMBC experiment to 8 for each increment. The HMBC experiment will take 30 minutes to collect, and we will resume the video after it is completed. This is the proton spectrum of the ibuprofen sample. It shows a peak for the carboxylic acid proton at 10.7 ppm. The para-substituted aromatic benzene ring has 2 pairs of chemically equivalent protons that fall between 7.1 and 7.3 ppm. The aliphatic region shows 5 chemical shifts that is consistent with ibuprofen. Based on integration, the isopropyl methyl protons are the doublet near 0.93 ppm and the other methyl is the doublet at 1.53 ppm. The heavily split isopropyl methine is the 9-line multiplet at 1.88 ppm. The doublet at 2.48 ppm, with the relative intensity of 2, is the methylene, and the quartet at 3.74 ppm is the methine that is adjacent to the carboxylic acid. This is the HMBC experiment for ibuprofen. Like the HSQC experiment, the proton chemical shifts, measured in F2, are along the horizontal axis. The F1 axis, running vertically down the screen, contains the C13 scale. At the top is the 1D proton spectrum in the F2 projection area. This trace was loaded by the automation from the spectrum run before the 2D experiment was performed. The projection along the F1 axis is the true shadow projection from the HMBC data. Its resolution is limited by the number of T1 increments, which is 128 by default for this experiment. Since the C13 window includes the 220 ppm chemical shift range, this comes out to about 2 ppm per data point. Also, the HMBC experiments collect magnitude mode datasets, and this will make the F1 resolution even poorer. If you have a 1D C13 spectrum available, you can replace the projection with this dataset. Right-click in the projection area and choose External from the pop-up menu. Enter the file name and experiment number that contains the 1D C13 spectrum and click on the OK button. This will make interpreting the results easier, but it is not necessary. As you can see, even for this relatively simple example, there are many cross-peaks scattered throughout the 2D spectrum. The easiest way to start interpreting the result is to examine the columns for each proton chemical shift and identifying the C13 that each cross-peak shows a remote coupling. Remember, these correlations are typically 2 and 3 bond couplings. In the aliphatic region, the intensity of the cross-peaks is similar for both the 2 or 3 bond cases. Just outside of the columns appear 2 peaks that form the doublet of the single bond correlation. These are usually lower than the cross-peaks for that proton resonance. However, the cross-peak intensity is proportional to the number of protons, so a suppressed single bond artifacts of one resonance could be close to the intensity of neighboring protons. To help interpret the results, we are going to use information we gained from the 1D spectra, COSY and HSQC results. Please review those videos to help follow along with the assignments made here. We are also going to use our knowledge of basic C13 chemical shifts. Let's start with the isopropyl methyl, which has a proton chemical shift at 0.933 parts per million. There are 3 cross-peaks that fall under this proton chemical shift. Since the C13 resolution is poor, we will use the chemical shifts from the 1D carbon spectra for a more precise value. The first correlation is at a carbon shift of 22.41 ppm. This represents the 3-bond coupling to the other methyl carbon of the isopropyl group and not the 1-bond coupling. It is common to draw out the HMBC correlations on the chemical structure like shown here. You will see such diagrams in journal articles. However, you need to remember that the correlations are through the bonds between the 2 nuclei. Next is the coupling to the carbon at 30.18 ppm. From the HSQC result we know that this is the carbon that the isopropyl methine is bound to. This is a 2-bond correlation. The third correlation falls at 45 ppm. As we saw in the HSQC result, there are 2 very closely spaced C13 resonances in this area. One was at 45.06 ppm and the other was 44.99 ppm. With a digital resolution of 1.7 ppm in the F1 dimension, there is no way we can distinguish which carbon is contributing to any of the correlations in this area. To achieve the F1 resolution of 0.07 ppm, we would have to run more than 1000 T1 increments. This would take more than 4 hours compared to the 30 minutes this data set was collected. However, we can use information from our other datasets and the chemical structure to know that the methylene carbon at 45.06 ppm is the 3-bond correlation seen here. The next proton we will focus our attention on is the isopropyl methine. It shows the expected 2-bond correlations to the methyl at 22.41 ppm and the methylene at 45.06 ppm. It also shows a crosspeak at 140.88 ppm. This chemical shift is consistent with an aromatic carbon bound to a methylene carbon. The methylene protons at 2.482 ppm show 4 correlations. As expected, we see the correlations to the isopropyl methyl and methine carbons. The 2 cross peaks in the aromatic region are to the chemical shift for one of the protonated carbons at 129.41 ppm, which is 3 bonds away. The other is to the non-protonated carbon at 140.88 ppm, that is 2 bonds from the methylene protons. We can now jump over to the other side of the aromatic ring and examine the methyl doublet at 1.533 ppm. The first of the 3 correlations is to the methine at 44.99 ppm. The HSQC and COSY results allow us to distinguish this from the other carbon in this area. The methyl also shows a cross-peak to the carbon at 136.97 ppm. This chemical shift is consistent with the value expected for the aromatic carbon that is bound to the methine. The third chemical shift is to the carbonyl carbon of the acid group that has a value of 181.07 ppm. The methine proton at 3.741 ppm shows the expected correlations to the methyl at 18.01 ppm, non-protonated aromatic carbon at 136.97 ppm and the carbonyl carbon at 181.07 ppm. It also shows a 3-bond correlation to the protonated aromatic carbon at 127.30 ppm. As you can see, we have made the complete assignment of the carbon chemical shifts from the HMBC data with a little help from the HSQC, COSY and 1D spectra. The aromatic protons should also be examined. The proton at 7.137 ppm shows 3 cross peaks. Note that these are all 3 bond correlations. First is to the methylene carbon at 45.06 ppm. Next is to the carbon at 129.41. This is the 3-bond, not the 1-bond correlation. The last is the correlation to the carbon at 136.97 ppm. Note that it is very common to not see the 2-bond correlations on an aromatic ring. This is due to the very small magnitude of the coupling constants these two pairs have. The other aromatic protons also show 3 correlations. They include the methine at 44.99 ppm, the 3-bond carbon across the ring to 127.30 ppm and the non-protonated aromatic at 140.88 ppm. Again, the 2-bond correlations are not seen. The lack of 2-bond correlations make HMBC experiments difficult to interpret on structures with significant aromatic content. Great care needs to be taken because sometimes you will see weaker cross-peaks, that are due to coupling to carbons 4 bonds away, more often than the 2-bond correlations. This concludes our video on running and interpreting the HMBC experiment. Combined with information from the 1D spectra, COSY and the HSQC experiments, the HMBC results are extremely helpful for making chemical shift assignments and for piecing together unknown structures. Every researcher using NMR should be familiar with these experiments and use them for solving structures and making spectral assignments. Please see the staff if you have any questions about the HMBC experiment or any other application of NMR for your research projects. Thanks for watching.